So we are starting with the digestive system, the histology of digestive system now, and what we are going to talk about is the details of esophagus in the beginning, and then move on to the details of the stomach, also the histological pictures of small intestine as well as the large intestine, and finally we'll be looking at some details of the liver as well. So we are beginning with the esophagus. Esophagus, since it is a gut tube, it is going to have 4 layers, which is mucosa, and then you have submucosa, then you have the muscular externa, and then the adventitia. Only small part of esophagus is lined by peritoneum, so there is serosa, but only in the lower part of the esophagus. Mostly it is covered by connective tissue, adventitia. Now let us look at some histological picture of esophagus. You see, we have taken the transverse section of esophagus showing this open space, which is the lumen, and lumen is lined by the mucosa. Under mucosa we have the submucosa, and over that we will have the muscular externa, and most outside will be the adventitia. Now we are going to magnify the picture, and you see this is towards the lumen side, the mucosa. As we understand, the mucosa, which is lining the luminal surface, will have some epithelium, which is stratified squamous epithelium, non-keratinized. So it is non-keratinized stratified squamous epithelium. What is this epithelium supported by? That is the lamina propria, some connective tissue, sub-epithelial connective tissue. And what is still it is being supported by? Some muscle of the mucosa, which is called muscularis mucosa. So mucosa is 3 components basically. There is the epithelium supported by subepithelial connective tissue, the lamina propria, and then you have muscle of the mucosa or muscularis mucosa. Then what is under the mucosa? If this is the mucosa, under the mucosa you have submucosa, and if that is submucosa, what is over the submucosa? That is the muscular externa. Some smooth muscle cells will be there. Now remember, submucosa is the strongest layer of the esophagus. And since it is the strongest layer of esophagus, it is to be included in the sutures. You know, whenever you are doing suturing, your suture should include the submucosa, because it is the strongest layer of the esophagus. And one more thing you find in the esophagus, the submucosa is having glands. This is a rarity to find glands in the submucosa if you are talking about the gut tube. Esophagus has glands in the submucosa, which is one peculiar finding. Though even in the proximal duodenum you have glands in the submucosa. Otherwise in the GIT it's rare to see glands in the submucosa. Glands should be in the mucosal area, not the submucosal area. Anyhow, as you are going to talk about the stomach, let us see the glands of the stomach. We want to focus upon glands of the stomach. Obviously, they will be in the mucosal area. Now, stomach is going to start with cardia region, and there is a fundus region and body and pylorus. We have to focus upon the beginning and ending. See, the cardia, which is the beginning of stomach, and the pylorus, which is ending of stomach, should have more of mucus secreting cells. Why the cardia and the pylorus should have more of mucus producing cell? Because you have to neutralize the acid which is being produced in the body. So which cells are producing acid in the body of the stomach? Parietal cells. And these parietal cells, when they are secreting acid in the body, there is a tendency of that acid to move into the lower esophagus and causing some ulceration, or maybe this acid will move into the proximal duodenum causing duodenal ulcer. You don't want duodenal ulcer, you don't want esophageal ulcer, so you're neutralizing the acid. Actually, my point is, in the cardia region and in the pyloric region should be more of mucus secreting cell. That means in the cardia and the pylorus you have less of the parietal cells. This came as a question. Parietal cells are rarely found in the cardia and pylorus because it will have more of mucus cells. Now, as you are moving further, you will see this question. It is asking the same thing. It is asking what is the exception for the gastric glands, and what do you think? You see, immediately you will notice that the choice number A is the answer. Why? Because we just mentioned that if it is about the pylorus or maybe the cardia, they will have more of mucus secreting cells, not the acid. So where is the acid producing cells then? Parietal cells, mostly predominantly found in the body portion. So what is there in the pylorus or cardia? It is more of mucus cell. So choice number B is okay. What about choice number C? That is correct. Chief cells are secreting enzymes like pepsinogen and mostly found in the fundus region of the stomach. Though any cell can be anywhere, but predominantly if you want to search for the chief cell, chiefly found in the fundus region of the stomach. And what are the parietal? Parietal cells not only secrete the HCl, but they also secrete the intrinsic factor for absorption of vitamin B12. Now, once you have this basic idea, keep your answer as choice number A. We are moving on to the gastric glands having some different type of cells, which we just named. See, this is the gastric lumen having gastric pit, having gastric gland, and in the gastric gland you have some cells like you have mucus cell. Do you remember that stomach do not have any goblet cell? This is not goblet cell. Stomach do not have any goblet cell. It is some other type of mucus cells. So there will be some surface mucus, neck mucus, and then there is a parietal, chief, and there is also endocrine cells. We want to talk about these glands in detail because there are questions upon that. So you see, there is a surface mucus cell and there is a mucus neck cell. Then there is a parietal cell which is secreting not only the hydrochloric acid but also secreting intrinsic factor, which we have just mentioned. If you're talking about the chief cell, they are secreting the enzymes like pepsinogen. And what about the neuroendocrine cell? The name is telling. Neuroendocrine cell will give endocrine hormones like the gastrin hormone is given by these cells. Remember one thing, if you are looking at the upper half of the gland, then you have this parietal cell and neck cell. If you're looking at the lower half of the gland, then you have chief cell and the endocrine cell. So there is upper and lower half, you can say, of the gastric gland. In the upper portion of gastric gland, you have mucus cell and the parietal cell, and the lower half you have the chief cell and the entero or neuroendocrine cells. How do you know which cell is which? Understand that parietal cell is pink color, PP. Parietal is pink. Why it is pink? Because it is eosinophilic. And it is the chief cell which is blue color. Why blue? Because it is basophilic. It is taking more of hematoxylin. So chief cell is taking more of hematoxylin, and because it is taking more of hematoxylin, blue color, basophilic. And who is taking more of eosin? This parietal cell. So parietal cell will be pink color because taking more of eosin. We will see some more details of this. Actually, we got a question. It is asking us which of the marked cells is secreting HCl. Obviously, they are asking about the parietal cell. So where would you find the parietal cell? Let us magnify the diagram. Let us magnify the upper part of the gland, and as you magnify upper part, you will find that this area is showing the mucous neck cells, and it is also showing the parietal cells. How do you know they are parietal cells? First of all, they are in the upper half of the gland, and number two, they are actually pink color. And not only they are pink color, you have to understand they are actually cuboidal cells, and they have central spherical nucleus. It is appearing like a fried egg appearance. So you are trying to tell, first of all, parietal cells are in the upper half of the gland. Yes, and then they are pink color because they are eosinophilic. Yes, and they are having a central spherical nucleus, means cuboidal cell, right? And they have this fried egg appearance, like this is the central yolk and that is the peripheral albumin. Fried egg appearance. Central yolk and peripheral albumin like appearance. Now, if you say so, fine. What about the lower half of the gland? This was the upper half. What about the lower half? If you want to talk about the lower half, let us go there. And when you are going to the lower half of the gland, again, you see, though there is more of the chief cell, but there can be also parietal cells. So upper area is parietal, lower is chief cell. How do you know that it is chief cell? As we mentioned, that the chief cells are going to be basophilic. So they are more blue in color. They take more of hematoxylin. Now, if you say comparatively, the chief cells are basophilic or taking more of hematoxylin and less of eosin, what about the parietal cell? You see, this is one parietal, another parietal. This is all parietal cells. So parietal cells are pink in color. So parietal are pink in color. Parietal are in the upper half of gland, chief are in the lower half of gland. So whatever you see here, this is all the parietal cells secreting HCl. That was our question. Who is secreting HCl? These cells, because these cells, chief cells, are secreting the pepsin enzyme. So this is our question. Let us look at the question again. It is asking who is secreting HCl. Our answer is parietal cell. So where is parietal cell? You have to understand, marker A is the surface mucous cell, and marker B is the mucous neck cell, and marker C, that is the parietal cell. And what is marker D? That is the chief cell. So D is the chief cell, dark color, and C is the parietal cell, light pink color, and B is the mucous neck cell, and A is the surface neck cell. So you mean to say the answer is choice number C? Yeah, choice number C, because choice number C is marker C is the parietal cell. That is how you identify it. Now, as you are talking about these different type of cells, which could be a surface mucous cell or a mucous neck cell or parietal cell or chief cell or endocrine cell, let's make the diagram for them now. And as you are making the diagram, what you show is, this is the gastric lumen, and lined by some gastric epithelium making the gastric gland. So this is gastric lumen lined by gastric epithelium and making the gastric gland. And this gastric gland was secreting, there was some secretions. Where are those secretions going? They will go to the gastric pit, and from the gastric pit it will go to the gastric lumen. So as we are telling, there are some secretions given by the gastric gland. They will go towards the gastric pit and then go towards gastric lumen. It is below upwards. What is the junction of the gastric pit and the gastric gland called as? That is called as the isthmus. Isthmus means junction. So what is isthmus? It is a junction. Junction of the gastric pit and the gastric gland. Now, as you're talking about the isthmus, what is below that is the neck region of the gastric gland, and then body of the gastric gland, then fundus of the gastric gland, or you can say base of the gastric gland. So we have isthmus, and then below that is neck region, body region, and fundus or base of the gastric gland. Which type of cells are there? You have to remember, if you are talking about the surface here, there will be a surface mucous cell, And what is in the neck region? Neck, mucus cell. And what is in the body region? Predominantly parietal cells. So parietal cells are predominantly present in the body region. Actually, upper part of the gastric gland has mucous neck cell and the parietal cells. And what was the parietal cell doing? Parietal cell was actually giving us two secretions. One of that was HCl, the other was intrinsic factor. And what is in the lower half of the gastric gland? In the fundus base region, we have a cell which is having lot of granules. This is called as chief cell, and chief cell is having lot of granules. Those granules are called as zymogen granules, and in the zymogen granules you have the pepsin enzyme. So the point is, the chief cell is at the fundus or base of the gastric gland and is having some zymogen granules, having some pepsin enzyme. It has three names. This cell has three names. You can call this cell as chief cell or zymogen cell or peptic cell. You call it as chief cell, zymogen cell or peptic cell, and it is at the base. And near the base or fundus you have one more cell which is called as neuroendocrine cell. And as the name is suggesting, this neuroendocrine cell is going to give you some endocrine hormones, maybe like the gastrin hormone. So when you're talking about gastrin hormone, it is given by neuroendocrine cell, and it is also near the fundus or base of the gland. Now there is one cell which is at the isthmus region. And which cell is there at the isthmus region? We are calling it as the stem cell. Now when you're talking about the stem cell, which is you are telling is at the isthmus region, is it only at the isthmus region? No, it is not like that. You have to understand, predominantly it is at the isthmus region, otherwise it can be at other places also. Actually, any of these cells can be anywhere, but they have some predominance. Like we said, parietal is upper and chief is lower. So isthmus has a predominance for stem cells. And what is stem cell for? Stem cell is for repair purpose. And what kind of repair? Say for example, there was gastritis and it is doing post-gastritis repair. Means in gastritis there might be damaged mucous neck cell or parietal cell or the chief cell, so you have to replace them, and that replacement is done by stem cell at the isthmus. It will migrate here, here and here and replace them, because the stem cell has pluripotency. It can repair the epithelium, post-gastritis repair. So these are all MCQs. In fact, now let us move on to the details of small intestine. And the small intestine is three parts. There is the duodenum, and then you have the jejunum, and then you have the ileum. How do you differentiate between the three? First of all, what are the common features? Common feature is that they are small intestine. So what is small intestine for? Increasing the absorption. And how do you increase the absorption? To increase the absorption of the nutrients, you have some things like plicae circularis. And what is plicae circularis? They are some mucosal fold to increase the surface area for absorption. Now, if you want to increase the absorption of nutrients, you will have plicae circularis, which will increase the surface area for absorption. They also have villi, finger-like projections, and at the microscopic level, they have microvilli. So small intestine, may it be the duodenum, jejunum or ileum, they will have plicae circularis and they have villi, microvilli. But remember, these plicae circularis, villi, microvilli are more in number in the jejunum, because towards the ileum it is decreasing. So plicae circularis will decrease and the microvilli will also decrease towards the ileum region. Jejunum has more predominant, because at the duodenum they are just beginning. So at the duodenum they are just beginning, maximum at the jejunum, then they are again reducing towards the ileum. How do you differentiate beyond this? Means what is the differentiating point? Let us see the details. You see, this is a diagram we have taken for duodenum and also the jejunum and also the ileum, and they have some common features like we have told earlier. Gut tube will have mucosa, and what is under that? Under that is some mucosa, and what is under that? Under that is muscular externa, and over that is serosa. So this is 4 layers of the gut tube which all time we have. Now, what is the differentiating point then? This is plicae circularis, mucosal fold. They increase the surface area of absorption, and there is villi also, finger-like projection. But as we suggested, maybe plicae circularis or the villi, microvilli, they are more in the jejunum and they are reducing in number towards the ileum. Ileum have less of villi and plicae circularis, though duodenum also has those features but maximized in jejunum. Duodenum has one feature, I told you earlier also, will have some submucosal gland, which is actually very rare to find. Having glands in the submucosa is a rarity, and you have seen that in the esophagus. You also see in the proximal duodenum. And what is the function of these submucosal gland here? Actually, they are called Brunner's gland. So, what is the function of the Brunner's gland then? We will find out what is the function of the Brunner's gland. They are present in the proximal duodenum, and they are submucosal glands, but they will be missing from jejunum and ileum. So, jejunum and ileum do not have glands in the submucosa. Ileum is identified by one fact that it will have a lot of lymphocytes, and they are called Peyer's patches in the terminal ileum. It is the MALT, mucosa associated lymphocyte tissue. Now, MALT is present everywhere, maybe duodenum, jejunum, ileum, but more pronounced in the distal small intestine, which is terminal ileum, so much that it is forming Peyer's patches. So Peyer's patches identify the ileum. So as you're talking about the names, who is this? Number 1, 2, 3, 4, 5, 6 in the small intestine gland. Number 1, enterocyte. What about the enterocyte? It is columnar cell. And what is this columnar cell doing? It is doing the absorption of the nutrients. So it is for the absorption of nutrients. What is number 2? It is the goblet cell. And what is the goblet cell for? Of course, secreting the mucus. And what is the third one? It is the Paneth cell. What is Paneth cell for? It is providing immunity. How is it providing immunity? It is killing the excess pathogen. And how will it kill the excess pathogen? Which pathogen are you talking about? Maybe bacteria or maybe protozoa like the amoeba. So it is killing the amoeba and the bacteria. Yes. How? How the Paneth cell will provide immunity by killing excess pathogen? How? What is the mechanism? Mechanism is that it will secrete some cytokines. And secreting those cytokines will kill the excess pathogen. It is to maintain the intestinal flora. You know, in the intestine, we need some microorganisms, but it should not be in excess. So, if you want to kill the excess of pathogens, some bacteria are required by us. We have some intestinal flora, but to maintain that intestinal flora, you have to keep the excess number, and that is by secreting cytokines by the Paneth cell. You will find that Paneth cell is identified by the fact that it is having apical eosinophilia. And why there is apical eosinophilia? There is apical eosinophilia because there are a lot of lysozymes at the apex. And what is inside the lysozymes? That is the cytokines. So Paneth cell is identified by the fact that it has apical eosinophilia because those lysozymes at the apex take a lot of eosin, dark pink apex, and those lysozymes are secreting the cytokines to kill the excess pathogen. What is cell number 4, 5, 6? Cell number 4, actually there is another immunity cell which is called M cell. What is that? Microfold cell. And what is microfold cell for? Cell number 4, which is M cell, microfold cell, is immunity providing. Actually, it is antigen presenting cell. It is picking up the antigen and presenting to the lymphocytes. What is cell number 5, 6? Cell number 5, 6 are universal cells. They are present in the stomach also. Did you notice stomach was having neuroendocrine cell to give some hormones? So there are some neuroendocrine cells giving hormones, and that will be seen in the small intestine. Even the stem cells, which are basically for repair purpose, were seen not only in the stomach, but they are also seen in the small intestine. So cell number 5, neuroendocrine cell, which is giving some endocrine hormones, and cell number 6, which is stem cell, which is for repair purpose, pluripotency, is not only present in the small intestine gland but also seen in the stomach. Now we want to look at some histological pictures of these cells, but at this moment you answer the question, which cells are not found in the small intestine? If you say which cell is not found in the small intestine, then I will definitely say stem cells are there, goblet cells are there, penis cells are there. So obviously answer is the next cell, which you have seen in the stomach, gastric gland. You have seen in the gastric gland, you have seen some mucous neck cell, but maybe it is not present in the small intestine. So we are telling that stem cell, goblet cell, penis cell may be there, but not the neck cell. That is your answer. Now once you got your answer, fine, but I want to look at the penis cell, some details. Not only penis cell, but the other cells also, like stem cell and the neuroendocrine cell. And for that purpose, we will go ahead. See, in the duodenum, we have found one thing. What was that? Specific for duodenum. See, duodenum will have villi to increase the surface area of absorption, which itself will have microvilli, forming some striated border also. Striated border means there are some microvilli forming, striated border. There are some intestinal crypts which will be forming the intestinal glands in the vicinity and duodenal glands. See, the thing was, there are some duodenal glands in the submucosa, and what do you call them as? The Brunner's gland. And what is this Brunner's gland doing? You wanted to know? Actually, Brunner's gland do two things by secreting one hormone called as the urogastron. So the point is, first of all, it's peculiar finding. You are having glands in the submucosa, and that is a feature of proximal duodenum, which is called as Brunner's gland, and doing two things by secreting hormone called urogastron. Urogastron is doing what? It will actually reduce the activity of parietal cell so that there is less of acid production. It is reducing the incidence of duodenal ulcer. And number two, it is going to increase the mitotic activity of the epithelium. What is the use of increasing mitotic activity of the epithelium? It is to heal the ulcer faster if there was some duodenal ulcer already present. So there can be some break in the mucosa, there can be duodenal ulceration. You want to heal it faster, so you will increase the mitotic activity. That is why urogastron is having one more name. It is called as epithelial growth factor, actually HEGF. So other name of urogastron is HEGF. It is epithelial growth factor, actually increasing the mitotic activity to heal the ulcer faster. So that is the job of urogastron. Actually, we got a question upon that. Let us see the question, then you understand it better. About the Brunner's gland, what is wrong? Are they in the submucosa? Yes. Are they secreting urogastron? Right. Which is going to inhibit the HCl production. How? By reducing activity of parietal cell, and then it is secreting HEGF. Yeah, HEGF actually is the other name of the urogastron only, because urogastron is increasing the mitotic activity to heal the ulcer faster. It is also renamed as HEGF. So you are trying to tell this is the answer. Yes. Why this is the answer? Because it is not the lower, it is the upper and proximal duodenum. So if it is the upper and proximal duodenum, why do you require the Brunner's gland in the upper proximal duodenum? Because that is where you have more chances of duodenal ulcer. You have more chances of duodenal ulcer in the upper and proximal duodenum. That is where you require the Brunner's gland, not in the lower duodenum. So keep this answer, choice number D, and proceed further. We will tell Brunner's gland are obviously in the submucosa, and they are secreting urogastron, which will inhibit the parietal cell, inhibiting acid production, reducing the incidence of the duodenal ulcer. And the other name of urogastron is HEGF, and which is increasing mitotic activity and heal the ulcer faster. But this statement is wrong, and that is the answer. It is not the lower duodenum, it is upper duodenum. Keep the answer as choice number D. Now, As you proceed further, this is a histological picture of the small intestine gland. So what do you find there? What I find there is the, you can say enterocytes. So where is the enterocyte? These are all enterocytes only. How do you know these are enterocytes? Because they are columnar cells. So there are some columnar cells which are enterocyte. What is enterocyte for? 1, 2, 3, 4, 5. They are for absorption of nutrients. And what is other cell? Other cell, we have this foamy appearance. Foamy appearance. What is this foamy appearance? Foamy appearance. Goblet cell. Here is the goblet cell. Here is the goblet cell. Let me magnify it. As you see, this is the goblet cell. It has foamy appearance. But why it is having foamy appearance? Because of the mucus. It is having some foamy appearance. Then what is in between? What is this? This is, of course, the enterocyte. This is enterocyte. This is enterocyte. Then what is this enteroendocrine cell? Enteroendocrine cell is the one which is giving us the endocrine hormones. So enteroendocrine cells are the one which will give us the endocrine hormones. And what is this Paneth cell? How do you identify Paneth cell? Paneth cell is identified by one fact that it is towards the base of the gland, the fundus of the gland, lower portion of the gland. Point 1. The Paneth cell is towards the lower part of the gland, base or fundus of the gland. And number two, it has apical eosinophilia. And why it is having apical eosinophilia? You remember? The apex has lot of lysozymes and they take lot of eosin. That is why. This is one Paneth cell, this is another one. So these are the three Paneth cells and they have apical dark pink apex. Dark pink apex. And why the apex is dark pink? Because of those lysozymes which were secreting the cytokines like tissue necrotic factor alpha. And why are you secreting tissue necrotic factor alpha? To kill the excess pathogen and maintain the intestinal flora. That is the job of the Paneth cell. It is immunity providing cell. So, Paneth cell is at the base, not towards the luminal villi. No, not towards the luminal villi. It is towards the base of the gland. And number two, it is having apical eosinophilia. So, these are the various cells in the small intestine which we wanted to find out. We already have seen some details. Let us go to the next thing, which is the large intestine. Now, when you're talking about the large intestine, I'll focus upon the cell types, which is very simple. There will be some colonocytes which are absorbing the water and electrolytes, also some vitamins, and they are also columnar cells. So there will be some columnar cells which are called as the colonocytes, and it is for absorption of the water, nutrients or minerals. But then there are also goblet cells, and remember the number of goblet cells is increasing towards the large intestine. Small intestine has less number, large intestine has more number, rectum has the most numbers, means most distal and more number of goblet cell to lubricate the fecal matter. It is to lubricate the fecal matter for easy passage of the fecal matter. So as the fecal matter keep going distal and distal, there is more of absorption of water, it will become dry. And because it is dry, it should be covered by lot of mucus. So goblet cell keep increasing distally and more so in the small intestine than in the large intestine, and towards the rectum it is maximum number just to lubricate the fecal matter for expulsion. That is the job. Now let us talk more about the anal canal, because there are questions on anal canal. You will see the epithelium of anal canal, but before that some gross anatomy. And you see, this is rectum, so there will be an anorectal junction, and then anal canal begins. When the anal canal begins, you will see anal canal itself has some zones. There is one columnar zone, and then there is anal transition zone. then there will be a squamous zone. So there is a squamous zone, anal transition zone, and there is a columnar zone. Yes. What is the boundary line? Actually, there is a dentate line, and then there is a pectineal line also. Line of Hilton. Yes, line of Hilton. So let us discuss these details by drawing a diagram ourselves. We'll draw a diagram ourselves now for the same. So what is your diagram talking about? Actually, 3 zones in the anal canal. And what are the 3 zones in the anal canal? We'll find that there is something called as the dentate line, which is also called as the pectinate line. Now, whether you say this is the dentate line or the pectinate line, what is it doing? It is the squamo-columnar junction, called as squamo-columnar junction, in fact. But not precisely it is. Why? Because below that is, of course, there is a squamous zone which is stratified squamous epithelium. Any external opening should have stratified squamous epithelium. But if you are looking at the upper regions, because we are telling that there is some anal transition zone, there you will have transitional epithelium. And what do you mean by transitional epithelium? That will draw a diagram and find out. But above the dentate line, you have anal transition zone. And what is this colorectal zone having? It will have columnar epithelium. There is a columnar zone or colorectal zone, or there you have the columnar epithelium. So this is the diagram which we are supposed to draw. Anal transition zone is called transition zone because what you find is the columnar epithelium will change into cuboidal epithelium, will change into squamous epithelium. Some transition will go on. What is that? Columnar epithelium, in the beginning, it was columnar epithelium, is changing into cuboidal epithelium, is changing into the squamous epithelium, and some transition is going on. That's why it is called anal transition zone. Let's just show it ourselves. So you are talking about anal canal here. Yes. And what is there in the anal canal? I have this line. What is this line called? This is called as dentate line, or you can also call it as pectinate line. Now, whether you say this is dentate or pectinate line, fine. What are the various zones you were telling about? Below that, I have the squamous zone. And what is above that? Anal transition zone. If that is the anal transition zone, then what is above that? Colorectal zone. Now, what kind of epithelium you have in the colorectal zone? I have the columnar epithelium. So, as you are telling that there are 3 zones in the anal canal, and they are more proximal, it is colorectal zone with columnar epithelium. Then you have the anal transition zone above the dentate line or pectinate line. Below the dentate line, pectinate line, you have squamous zone. Actually, you call it as squamous zone because here the epithelium is stratified squamous epithelium. That's why squamous zone. So below this line, I have the external opening, any external opening should be lined by stratified squamous epithelium. Then what is the epithelium here? Actually, there's a transition going on. What transition is going on? In the beginning, you have columnar epithelium, which is changing into cuboidal epithelium, which is changing into the squamous epithelium. So, in the beginning, you have some columnar epithelium, which is changing into cuboidal epithelium, is changing into squamous epithelium. So, some transition is going on. That is why we are calling it as transitional epithelium. So, transitional epithelium, which is in the anal transition zone, is above the dentate line, pectinate line. Yes. And there was one more line you were talking about. Which line is that? That line was actually white line of Hilton. And what about the white line of Hilton? As you are talking about the white line of Hilton, you will find it is anocutaneous junction. What is that? Mucocutaneous junction. What is that? This is the mucocutaneous junction because before that it is the anal mucosa. After that it is the true skin. You mean to say before that is anal mucosa? Yes. And after that there is a true skin. Yeah. Proximally you have anal mucosa, distally you have true skin. So this is called mucocutaneous junction. So what is the detail? Actually, this white line of Hilton will tell us some details like above this line is the pectine region. And what kind of epithelium in the pectine region? It is stratified squamous but non-keratinized because it is wet surface. You mean to say, below the dentate line and above the Hilton line, there is a pectine region which is wet surface? Yeah, and because it is wet surface, it is non-keratinized. And what is below the white line of Hilton? It is actually keratinized. So this will be keratinized stratified squamous epithelium. Yeah. Along with that, you also have sweat gland and sebaceous gland. Are you trying to say that the anal canal has sweat and sebaceous gland? No. What I'm trying to say is, anal canal does not have sweat and sebaceous gland until you are very much at the terminal area. So the most terminal area of the anal canal, which is below the white line of Hilton, which is keratinized epithelium, will become dry. You don't want a dryness, so that's why you have some sweat gland, sebaceous gland with the non-keratinized epithelium in the anal canal below the white line of Hilton. You'll find this is the question. What is the lining epithelium below the pectinate line or dentate line? What do you think is the answer? Of course, you have found your answer. It is non-keratinized stratified squamous epithelium. But do you realize that anal canal has all the four epithelia? Can you tell me one thing? This kind of epithelia is present in which zone? The most proximal colorectal zone. And what is this? It is actually present in the anal transition zone above the dentate line. And what is your answer? My answer is here. And what is the terminal most anal canal? Terminal most anal canal is keratinized, and not only keratinized, there is sweat gland and sebaceous gland also below the white line of Hilton. But our answer should remain choice number C. So keep your answer as choice number C, and let's proceed further. As we're moving further, we have to look at some details of liver now, and you magnify the histological picture of liver. As you're talking about the liver, you have taken a specimen from here, and you find that liver is having dual blood supply. There is some oxygen brought by what is called as the hepatic artery, and there is some nutrients brought by what is called as the portal vein. So what is this? This is actually portal triad, and in the portal triad you will find that there is a branch of hepatic artery bringing the oxygen, and then there is a branch of portal vein bringing the nutrients. And what is this green color for? Actually it is DAB structure. And what is DAB full form of D? D is the duct, bile duct. And what is the A? A is the artery, hepatic artery. And what is the V? V is the vein, portal vein. So can you see the three structures here? Three structures. So they are portal triads, basically, portal triad. So DAV structure is the portal triad. You have triad means three things. Yes. Okay. Now, when the portal vein brings the nutrients, it will be passing through the hepatocytes and going towards what is this? In the center. Because it is in the center, it is called as central vein. Let us still magnify this. We want to magnify this area, then more details can be discussed. So when you magnify this, you will find it here. We have a diagram, and what we are telling is, we have got this branch of portal vein which brings nutrients, and those nutrients are passing through hepatocytes. They will metabolize the nutrients, and the metabolites will be received by central vein, and the central vein will give it to the hepatic veins, and hepatic veins will give it to the inferior vena cava. So what is the circulation like? Intestine is absorbing the nutrients into the portal vein, into the hepatocytes, which will metabolize the nutrients and carry the metabolites to the central vein, which will then carry it to inferior vena cava. Whatever you are eating, the nutrients absorbed from intestine are carried by the portal vein towards the liver cells for metabolism. The metabolites are going into central vein, into the hepatic vein, into the inferior vena cava. And now you need some oxygen also, which is carried by the branch of hepatic artery. So branch of hepatic artery will bring the oxygen. And what are the hepatocytes doing? They will form some lymphatics also, and actually bile. Now, when the hepatocytes are forming bile, the bile will go into the bile duct. It is going into bile duct. So this is the DAV structure. D for duct, the bile duct. A for artery, the hepatic artery. V for vein, the portal vein. This is the portal triad. Now we have to still magnify the diagram because I want to see inside the hepatic sinusoids, which are fenestrated. What is fenestrated? Means they have windows. The endothelium is non-continuous. So you are going to look into the hepatic sinusoid, yes, and with fenestrations, right, and there is a break in the endothelium. Why break? Because there should be some transfer of contents from blood into the periphery of the hepatocytes. What is that space called? That is called the perisinusoidal space of Dece. Now, when you're telling it is the perisinusoidal space of Dece, I cannot see. This is quite, I have to magnify. This is quite small. Yeah, so let us magnify it and then you will discuss. See, this is the hepatic sinusoid and it is lined by endothelium, but the endothelium is fenestrated, means opening, 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 fenestration. So they are not continuous boundary line. So it is not a continuous boundary line. The endothelium which is covering this sinusoid, this is the sinusoid. The sinusoid which is having some endothelial lining is fenestrated. And why it is fenestrated? So that there is some passage of fluid from here to there. And what is this space around the sinusoid? That is called perisinusoidal space of Dece. Let's make a diagram for that. When you make the diagram for perisinusoidal space of Dece, there you will see a cell which is called the hepatic stellate cell of Ito. What is the function of the hepatic stellate cell of Ito? It is helping us to absorb the vitamins like vitamin A, vitamin D. So it is helping us to absorb vitamins A, vitamin D. Yes, hepatic stellate cell of Ito. It is in the perisinusoidal space of Dece. This is the diagram which we are going to draw. We also will show Kupffer cell. Where? Within the sinusoid. What is it? It is a derivative of monocyte for phagocytic function to pick up antigen. You see it is sending some pseudopodia. Where? Into perisinusoidal space. Why? To pick the antigens if there are some antigens. So that is Kupffer cell, a derivative of monocyte. It is a phagocytic cell. It is running in the blood, but sending some pseudopodia into the perisinusoidal space. Let's draw the diagram now. See, this is the sinusoid. But this sinusoid, when it is lined by this endothelium, this endothelium is not continuous. It is not continuous. It has breaks. And those breaks which you are seeing are called as fenestrations. So there are fenestration. There is non-continuous lining. And why do you need a non-continuous lining? Why do you need spaces like that? To let the fluid pass from here to there. Here to there means from where to where. Actually, this is sinusoid. It will have blood. Now, if you say this is sinusoid and it is having some blood, what is around that? What is that space called? Around that there is some space, and that space is called as perisinusoidal space of Dece. Now, what do you see in the perisinusoidal space of Dece? And what do you see within the sinusoid? First of all, this sinusoid is lined by some endothelium which is having some fenestrations. They're non-continuous lining, and those fenestrations allow some fluid to pass here and there from the blood into the perisinusoidal space of Dece. And what do you see in the perisinusoidal space of Dece? There I find the hepatic stellate cell. When you're talking about the hepatic stellate cell of Ito, what was this cell doing? As we suggested, it is helping us to absorb the vitamins like vitamin A, vitamin D. And what was inside the sinusoid? Inside the sinusoid, we were having the Kupffer cell. And what about the Kupffer cell? Kupffer cell is actually a monocyte derived lineage. And what is the monocyte derived lineage doing? You know, it is a phagocytic cell. So monocyte from the bone marrow will be traveling to the liver to become Kupffer cell, and it is a phagocytic cell. It will pick up the antigens and kill them. Means kill the antigens like that. That is their job. And they keep sending some pseudopodia here into the vicinity of the hepatocytes. So there are some hepatocytes here, and in the vicinity of hepatocytes you are having this space of DC, and that space of DC you have the pseudopodia coming from the Kupffer cell. So Kupffer cell sending pseudopodia for collecting some antigen which may be around the hepatocyte in the perisinusoidal space of DC. These are all MCQs in fact. Now moving further, how do you identify the slide of liver? By looking at the slides many times. Practice MCQs. That is the job. Practice MCQs. So what is this? This is the central vein. If that is the central vein, it will have some blood which is being given towards the hepatic veins. But where this blood came from? Because this is carrying the metabolites. Where the nutrients came from? Nutrients actually come from the, here you see, portal vein branch. So nutrients will be here in the portal vein branch, and the fluid will, the blood will, keep shifting towards the, this is the direction. So blood keep going through that area, and the hepatocytes keep metabolizing the nutrients, so that ultimately the nutrients will be given to hepatic vein. And what else do you have? I have a bile ductule which will be collecting the bile secreted by these hepatocytes. So these hepatocytes secrete bile and give it to the bile duct, and then I have some oxygen brought by this branch of hepatic artery. So branch of hepatic artery bring the oxygen. This is the DAV structure, the portal triad. Now there is a concept of lobules in the liver. We have to discuss that also. As you're talking about the liver units, actually there is liver acinus, liver lobule, all that is to be discussed. In fact, the old story is that there is a classic lobule, and which is based upon central vein. So it is having central vein in the center, and the blood keeps moving towards the central vein, and there is hexagonal structure, hexagonal. So the oldest is classic lobule, which is hexagonal, and it is having the central vein in the center. Blood keeps going towards that. And what is at the periphery then? Of course, the portal triads are at the periphery. This is old concept. Then came a newer one, which is portal lobule. Portal lobule was triangular and centered on the portal triad. So it was centered on the portal triad. This was the point number 2, second in number. First was classic lobule, then there was portal lobule. So portal lobule, which came next, was a triangular structure, and it is centered on the portal triad. What is at the periphery then? Sites. At sites you had central vein, and central vein. So you have 3 central veins, and they are at the sides, but the center was portal triad. Then came the third concept, which is liver acinus. This is the most widely accepted concept. So liver acinus is depending upon the blood which is carried by the branches of hepatic artery, portal vein. So liver acinus was centered on the blood vessels which are hepatic artery branches or portal vein, and it is giving some zones basically. And what is at the sites? Actually at the sites you have central vein on one side, and you have the portal triad on the other side diagonally. Let me magnify it further. You see, if you are talking about the oldest classic lobule, which is the very first one to come, it was telling that structure is hexagonal and the blood is draining towards central vein. But then came the next one, which was portal lobule. It was based upon the drainage of bile into the portal triads, and at the center was portal triad. It was a triangular thing. Center was portal triad and bile drainage. In the periphery, central vein, and central vein. And what is the latest one? Third one is portal acinus, which is based upon the branches of hepatic artery and the portal vein. It is actually telling us about the zones of ischemia or toxic injury. What is the zone of ischemia, toxic injury? What is it about? Actually, see, this is some branches of hepatic artery, portal vein, and near that is high level of oxygen, beyond that low level, and still distal the least level. So this area is highly oxygenated because that is where you have the blood vessels passing, and then the other area will be less oxygenated and this area will be least oxygenated. And moreover, one more thing, if toxins are coming in the blood, if toxins were coming in the blood, then you will find that more affected area is here. So this is the more affected area by the toxins, this is less affected, this is the least affected. So this is, depending upon the oxygenation and the toxic distribution of the structures which were carried by the blood vessel. On one end it was portal triad, and on the other end it was central vein and the central vein. Liver acinus.